Welcome to our lecture on Chapter 6, everyone. The title of Chapter 6 is Metabolism, and the lecture on this chapter is going to be the first stepping stone on a journey that will continue over the next two chapters as well. Chapter 7 and Chapter 8 are going to build on what we learned here today. And where I like to start this journey is by asking you to consider what you had for breakfast this morning. You may have eaten any number of foods, eggs, toast, yogurt, fruit, cereal with milk. But regardless of what you ate, what all of these foods have in common is that they are all composed of the nutritional macromolecules that make up living organisms: carbohydrates, lipids, and proteins. Because all of these things come from living organisms. Apples come from trees, bread comes from wheat, eggs come from chickens, milk and yogurt come from cows. Everything that you see here was sourced from something that was alive, and therefore they are made of these macromolecules. And when you eat these things, your body's goal is to obtain energy from those macromolecules. This gets back to one of the properties of living things that we learned about back in Chapter 1, energy processing. As you know by now, all living things obtain and use energy. Whether you are an animal, a plant, a microscopic single-celled bacterium, every living thing needs energy. And this chapter is going to get into a bit of the mechanics of how living things get that energy. But before we dive into those mechanics, let's think about why living things need energy. Why do we need energy? This might seem like a really elementary question, but let's get specific in pinpointing things that our bodies and cells need energy for. In fact, this will be your first checkpoint task of the lecture. I want you to think back to our previous lecture on Chapter 5. In that chapter, we learned about a specific transport process that requires energy to carry it out. What was the name of that process? Now, this is just one very small example of something that cells need energy for. There are so many different reasons why cells need to process energy, including building new structures and organelles, reproducing new cells, which we're going to cover how that works in our next unit, repairing damage to the cell, clearing waste materials out from the cell, defending against invaders like viruses. In some cases, moving the entire cell. Not all cells are mobile, but some do move around and they require energy to do so. Responding to environmental signals, and much, much more. So to really just suffice it to say that energy processing is very important. It's a very important aspect of what it means to be alive and everything that entails being alive. So much so that much of an organism's metabolism is dedicated to extracting, storing, or utilizing energy. But what is metabolism? Metabolism is a word that you've probably heard before. And in the context that you've heard it before, you might associate it with people talking about how fast or how slow their body digests food, how efficient it is at extracting energy and storing it. This is true. Those digestion processes are metabolism, but metabolism is also much bigger than just those processes. The actual definition of metabolism is all of the chemical reactions that take place inside of organisms and sustain their livelihood. Metabolism is basically everything that happens on a chemical level inside the body of a living thing. So it's a very, very broad, encompassing concept. And being so broad, it's helpful to break it down and think about metabolism as being composed of two interdependent parts. First, there are anabolic reactions, which take smaller molecules and combine them to create larger ones. These types of chemical reactions require energy to move forward, which makes sense, right? Imagine you're building a house. A lot of energy goes into the process of combining the smaller construction materials to create the larger final product. That is an anabolic reaction. And then the other half of the coin is that there are catabolic reactions. This is the opposite. These reactions break down large molecules into small ones, and they release energy in the process. So think about a fire, for instance. When you burn a piece of wood, when you burn a log, the reaction of the fire takes the wood and breaks it down into smaller pieces. And in the process, a lot of energy ends up getting released. So we've got anabolic and catabolic reactions. And my easy peasy recommendation for remembering the difference between the two is to think first about anabolic steroids, which are used to build muscle, just like anabolic reactions build up larger molecules. And then for catabolic reactions, think of cats. Cats love to destroy things and break them down. So catabolic reactions are ones that break down large molecules into smaller ones. So to practice this, let's think about another example. Think again about whatever it was that you ate for breakfast this morning. When your body digests that food, is that going to be an anabolic or a catabolic reaction? Okay, so getting back to metabolism, we have reactions that build up and we have reactions that break down. And energy is an integral part of each of them. Energy is either being consumed or released in virtually every chemical reaction. But to really understand metabolism and how these reactions take place, we need to learn a little bit more about energy itself. Firstly, what is energy? In science, energy is defined as the ability to do work. And energy can exist in two different states. Kinetic energy is one of these, and that is the energy of motion. This means energy that is actively engaged, energy that is currently and presently being used. Potential energy is the second form, and this is stored energy, energy that is not currently in action, but it has the potential to be. One example that shows the difference between potential and kinetic energy is water that's flowing over the edge of a drop off, like what you see here going on with this dam. The water at the top has potential energy because it has the potential to be in downward motion, while the water that is actively falling over the edge has kinetic energy because it is currently and presently in motion. From this example, we can probably also see that energy can be converted between these two states, meaning that potential energy can become kinetic energy and vice versa. When that water at the top of the dam starts to fall over the edge, now all of the potential energy that it had, the stored energy, has been turned into active, moving kinetic energy. And this is just one way that energy can be stored. There are many different types of potential energy. The example that we just looked at is called gravitational potential energy, where an object is suspended and it has the potential to fall toward a center of gravity. But there is also elastic potential energy. An example of this is a rubber band that is stretched out. So it's not in motion, it's just holding position, but it has the potential to be in motion because it is poised to snap back with that elastic force. But the type of potential energy that we are interested in as biologists is not either of these. It's a different type called chemical potential energy. Chemical potential energy is the energy that is stored in molecules. And unlike gravitational potential energy or elastic potential energy, we can't see chemical potential energy. But we know it's there, right? We know that chemicals contain energy. And that's why eating food, which is made of chemicals like carbohydrates, lipids, and proteins, that food is able to supply us with energy. So, as we said before, one of the big goals of the body's metabolism is to extract energy, specifically by taking the chemical potential energy and converting it into kinetic energy. The kinetic form is what allows us to function. Okay, so how can the energy that is stored in those molecules be extracted? We know that gravitational potential energy transforms into kinetic energy when it falls towards a center of gravity. We know that elastic potential energy becomes kinetic when the elastic force causes the material to snap back. So, how does chemical potential energy become kinetic? To answer that question, we need to understand that the chemical potential energy in molecules is stored in their chemical bonds. I'll say that one more time just to drive it home. The potential energy found in chemicals is stored in the bonds between the atoms within those molecules. And you think about it, this makes sense because what are chemical bonds composed of? They are made of electrons. We spoke previously in chapter 2 about how there are covalent bonds where molecules share electrons with each other. That's what you're seeing here on this slide. There are also ionic bonds where electrons are transferred. But regardless of the type of bond, the action centers all around the electrons. The electrons are what carry the energy. And if you think about how electricity works, like the electricity that runs our houses and our buildings, it is electrons found in the metal of the wires that carries the electric current, which is how the energy of electricity gets transmitted. So just remember that electricity comes from electrons. And if you remember that, then it makes perfect sense why it's the electrons in the chemical bond of a molecule that are the epicenters of where you find all of the energy. Another thing to realize is that not all chemical bonds are equal in the amount of energy that they store. It depends, actually, on which elements are involved in the bond. For example, here we have a molecule of glucose, which might look familiar to you from back in chapter 3. Glucose has a number of different pairs of atoms bonded together within its structure. One pair that you'll see bonded in a few different places in this molecule is a carbon atom and a hydrogen atom. A covalent bond between a carbon atom and a hydrogen atom contains 98.9 calories of potential energy per mole. And you don't really need to know this for our class, but a mole is a unit of measurement commonly used in chemistry that is equal to a specific number of molecules. For comparison, a bond between a carbon atom and an oxygen atom, which you can also see happening in glucose, contains just 85.6 calories of potential energy. And then a bond between two carbon atoms contains 82.9 calories of potential energy. So the key point here is that bonds between different atoms store different amounts of potential energy. And because of this variation, different molecules also have different total amounts of energy. So let's look at the energy level stored in each of these two molecules for comparison. But before we do, a checkpoint question. Think back to chapter 3. Each of these molecules is a monomer or a building block of a larger molecule. Based on their structure, what is the name of the macromolecule that you could build from each of these? What is the macromolecule for glucose? And then what would be the macromolecule for lauric acid? So the bottom line is that different bonds have different energy levels. And because different molecules have different structures and different numbers and types of bonds within them, these different molecules also have varying total amounts of energy stored in them. For example, 1 ounce of glucose contains about 113 calories, but an ounce of lauric acid, which is the one on the bottom, an ounce of that stuff contains 265 calories. That's over twice as much as what's contained in an equivalent amount of glucose. So different molecules have different amounts of stored potential energy. Okay, so we know where and how the energy is stored in these molecules. But what good does that do us? We need to know how we get the energy. How do we extract it? It's sitting there in the form of electrons in a chemical bond. So how do we actually make use of it? Well, the way that the energy can be harvested from a molecule is by breaking the chemical bonds so that those electrons and their energy can be moved around and accessed. And this is what's known as a chemical reaction. A chemical reaction occurs when a molecule's bonds are broken or rearranged. And then when those bonds are broken and the energy is released, it can be transferred to a more easily accessible form. Because the reality for cells is that molecules like carbohydrates, like lipids, like proteins, they have a lot of stored potential energy, but that energy is not very easy to access when it's packaged in the form of these large, clunky molecules. There is an alternative, though: adenosine triphosphate, or ATP. ATP is often described as the energy currency of the cell because not only does it store large amounts of energy, it's a small molecule that can be easily moved around the cell, and it stores this energy in a universally accessible way. We can think about carbohydrates, fats, and proteins as being like gold bars or precious jewels. These things have a lot of value, but you can't walk into a store and hand someone a gold bar and expect to use it as currency for your transaction. ATP is like cold hard cash. It has value and it's extremely usable for all of the cell's energy needs. So, if we take a look back at this slide, we really should modify it a bit and show that one of the major goals of metabolism is to take chemical potential energy in its not accessible form, the macromolecules, convert it to ATP, which is the more easily accessible form, and then that form is the one that can be put into action as kinetic energy. Now, let's briefly take a closer look at this molecule known as ATP. If it looks a little bit familiar to you, that is not a coincidence. ATP does bear a striking resemblance to another molecule that we learned about back in chapter 3. I'd like you to tell me what that molecule is. And these two molecules are not fully identical to each other, but they do share a very similar structure in that they have three separate parts. They have a sugar, a nitrogenous base, and they have phosphate groups within them. What molecule that we learned about in chapter 3 has a sugar, nitrogenous base, and phosphate group? Okay, so what is it that is special about the chemical structure of ATP that makes it capable of storing lots and lots of energy? You may notice on the left-hand side of this molecule, what's shown in yellow, there are three phosphate groups attached to each other in a line. And that's where this molecule gets the part of its name, triphosphate. Triphosphate means three phosphate groups. You can see the three P's surrounded by O's, oxygen. You may also notice that the oxygen atoms in these phosphate groups, they each have a negative electrical charge. Those negative charges are the key to ATP's energy potential because the negative charges repel each other just like two magnets of the same pole. Have you ever tried to press two magnets together and they're trying to force themselves apart? Negative charges repel each other just like magnets do. And therefore, keeping these three phosphate groups bonded together takes a lot of energy. It requires a lot of force, just like it requires you to expend a lot of energy to hold two magnets from the same pole together. They want to fly apart. And so the bonds that hold them together have high amounts of chemical potential energy. And in fact, that's exactly how the chemical potential energy stored in these bonds between those phosphate groups is released. The high energy bonds between the second and the third phosphate group get broken, and that allows the energy that's in that bond to be released and transferred. And when this happens and the third phosphate group is lost and separated, the molecule transforms into adenosine diphosphate, ADP, instead of adenosine triphosphate, ATP, because now, once it loses that third phosphate group, it only has two, di meaning two. So we can think about ATP that you see on the left as being like a fully charged battery. And when that bond is broken and it's transformed into ADP, the battery then loses its charge. However, the ADP molecules can be recharged back to ATP, the fully charged form, by simply adding another phosphate group back into the chain. So this is what we're going to see happening as we get into our next two chapters, chapter 7 and 8, and we journey through the process of cells obtaining energy. We'll see molecules of ADP being recharged into molecules of ATP, using the chemical energy stored in the nutritional macromolecules. So stay tuned for that in our next 2 chapters. So at this point, we're going to take a moment to recap what we've talked about so far, because this chapter is pretty technical and abstract, so it's important to step back and look at the big picture. At this point, we have established that all living things need energy to function, and that energy has 2 forms: kinetic energy, which is the energy of motion, and potential energy, which is stored energy. Living things need to do all sorts of kinetic energy activities, and they have to derive that kinetic energy from some stored source, which turns out to be chemicals. Living things get their kinetic energy by extracting the potential energy stored in chemicals. How they do this is by performing chemical reactions that break and rearrange the bonds in molecules in order to get at the electrons, which is where the energy is actually coming from. And it's important for living things to be able to shuffle these bonds around, because the potential energy found in macromolecules like carbohydrates and fats is not very accessible. So they have to take that energy and move it into a different storage unit called ATP, which is the cell's main energy currency. And when stored in the form of ATP, the energy can then be used to make other chemical reactions happen within the cell. Now, if all of that makes sense to you, you have all of that straight in your head, let's move on to the next question that we need to address. So we've just said that the energy stored in ATP can be used to make other chemical reactions happen within the cell. But do all chemical reactions require energy to happen? Does every little step or process that happens in your cells need a burst of energy to power it? And the answer is no. Some chemical reactions actually happen spontaneously. Let me give you an example. The breakdown of sucrose, which is the chemical that we know as regular granulated table sugar, this is a spontaneous reaction that actually happens on its own without needing any energy behind it to fuel it. So that's the molecule that you see on the screen on the left. That molecule is the structure of sucrose. And it will spontaneously break down into the building blocks that compose it, which are the monomers glucose and fructose. Now, before we go any further, let's do another checkpoint to review. Think with me back to chapter 3. Would you classify sucrose, that is the original table sugar, as a monosaccharide, disaccharide, or polysaccharide? Feel free to rewind a couple of slides and look at its structure. And then what about glucose and fructose, the 2 building blocks that make up sucrose? Are those monosaccharides, disaccharides, or polysaccharides? So we've just established that some chemical reactions require energy, others do not. So the question that emerges from this is why? Why is that the case? Why do some chemical reactions require energy and others don't? And the answer to this, a very high-level summary, is the laws of thermodynamics. You may have heard or learned about the laws of thermodynamics before, and this isn't a chemistry class or a physics class, but we still want to see how this applies to biology and the energy processing that happens in living organisms. So let's take a look at how the laws of thermodynamics control whether or not something happens spontaneously or not within living organisms. Our first stop is, naturally, the first law of thermodynamics, which states that energy is never created or destroyed, it only changes forms. To put this another way, what this means is that the total amount of energy should be the same before and after a chemical reaction takes place. So if we were to look at the amount of energy stored in a molecule of sucrose, which you can see on the left side of my little scale here, and then compare it to the energy after it undergoes breakdown into glucose and fructose, which you can see on the right side of the scale is balanced because the 2 amounts of energy should be equal. Okay, that's simple enough. But the key really lies in the second law of thermodynamics, which states that there is a universal tendency for disorder, which in thermodynamics is also called entropy, to increase. There's a universal tendency for disorder and disorganization to increase. In other words, if a chemical reaction causes higher levels of disorder, it will happen spontaneously. Think about it this way: there is a natural tendency for your clean room in your house to become messier and messier over time. The entropy increases. Of course, we're talking about disorder increasing on a molecular level, but the messy room is still a nice analogy. So, energy has to be the same before and after a chemical reaction. And if disorder increases, the reaction happens spontaneously. The final key, the final piece of information to put this all together is knowing that heat is the most disorderly type of energy. If you were to line up different types of energy on a scale from less disorder to more disorder, you would find heat at the far end of the spectrum with the most disorder. Other types of energy, think of radio waves, visible light waves, X rays, chemical potential energy, all of these are less chaotic and more orderly than heat energy. And what this means is that converting any other type of energy into heat causes an overall increase in disorder. I'm going to say that one more time because that's really the key. Converting any other type of energy into heat causes an overall increase in disorder, which, according to the second law of thermodynamics, is something that has a natural tendency to spontaneously happen. So, getting back to our example of the breakdown of sucrose, why does this chemical reaction happen spontaneously? It's because when sucrose gets broken down into its two monomers, glucose and fructose, some of the stored chemical potential energy gets converted into heat. It goes from being this nice, orderly form of chemical potential energy into being heat energy, which increases the overall amount of disorder here. Another way to look at this, if we want to draw again on our little scale that we used when we were talking about the first law of thermodynamics, is that we start off with a certain amount of chemical potential energy in a sucrose molecule. That's what this big green block represents. That's the amount of chemical potential energy found in a molecule of sucrose. And by the end of the chemical reaction, of its breakdown, you have two different types of energy. You have the chemical potential energy found in the building blocks, glucose and fructose. That's the smaller green block on the right-hand side, as well as some heat energy that was released, represented by the red block. As you can see, the total amount of energy is the same before and after the chemical reaction. The scale is still balanced, which means we're in compliance with the first law of thermodynamics. Energy is never created or destroyed. But on the after side, the right-hand side, some of the energy has become heat. And why is this significant? It's because some of the chemical energy turned into a more disorderly form. And according to the second law of thermodynamics, which is that there's a universal tendency for disorder to increase, this is right in line with that tendency. So, as a consequence, this chemical reaction happens spontaneously. It happens on its own, without any help, without any nudging from external forces. The universe wants this reaction to happen because it results in more disorder. So, this is one example of a real life chemical reaction that occurs spontaneously and releases heat. But there are innumerable chemical reactions that happen inside of living organisms every single moment. And if those reactions are spontaneous, they generate an output of heat. So now I have a checkpoint question for you to think about, which is, what do you think happens to all of the heat that is given off by the spontaneous chemical reactions in your body? And I really want you to just sort of think about this, flex your critical thinking muscles for this question. If it helps, I suggest you put your hand on your forehead and notice Does your forehead feel warm? Why do you think that is? Where is that heat coming from, and where does it end up going? Okay, getting back to where we left off. We just explained how and why a chemical reaction is spontaneous, and what that has to do with the laws of thermodynamics. To reach the next level of understanding, we're going to introduce a new term. The energy in this diagram that you see represented by the green blocks, that chemical energy is referred to as free energy, specifically Gibbs free energy. Gibbs free energy is an umbrella term that is defined as any type of energy that is useful for doing work. Gibbs is the guy it was named after, and free energy just means energy that is free to use. So the chemical energy found in the sucrose before the reaction takes place is considered Gibbs free energy, and the chemical energy remaining in the glucose and fructose after the chemical reaction is also free energy. This is stored potential energy that is available to be used. On the other hand, the heat energy that is generated from the reaction is not considered Gibbs free energy. Heat energy is not usable energy. As you may have concluded in that last checkpoint we did, the heat just dissipates and diffuses away into the universe. It's not useful to make any processes happen in your body. So heat is not part of the umbrella term Gibbs free energy. Now, why is this idea of free energy important? It's because this is the standard way that biologists think about energy in biochemical reactions. It's in terms of how free energy ebbs and flows over the course of the reaction. And this flux of free energy gets mapped out by biochemists in a special type of graph called a free energy diagram. What you're looking at here is an example of a free energy diagram. This graph shows how free energy changes over the course of a chemical reaction. And if you're the type of person for whom graphs strike fear into your heart, don't worry, we are going to break this down and walk through it slowly. So let's get a closer look. And let's start with a blank graph, and then we're going to build it up together. So what we see here is the y-axis, the vertical one on the left side. This one is plotting Gibbs free energy. And the x-axis, the horizontal one along the bottom, that one is plotting time. So as you move from left to right across the graph, what it's showing is how the free energy changes as time passes. If the line goes up, that means that free energy is increasing over time. If the line goes down, that means it's decreasing over time. Okay, so now let's bring back that line that we had. So the path of this purple line starts at the beginning with the reactants. The reactants are the thing that undergo change in the chemical reaction. It's your starting point. If we're going to keep following along with the same example that we've been using, then the reactant in this case is sucrose. So let's drop a little sucrose molecule here to show exactly what we're talking about. As we now know, that sucrose contains a specific amount of stored chemical energy. So let's plop that on here too. That is our Gibbs free energy found in sucrose. When the chemical reaction begins and sucrose starts to break down, there is going to be this jump in energy where the free energy goes up, and then it slides back down to its final resting place at the products. We will talk about that jump in energy and its significance in just a second. But for the moment, what we really want to focus on is just the reactants and the products. Remember, the products of this chemical reaction are the two monomers that you get when sucrose is broken down, which are glucose and fructose. So here's our little glucose and fructose molecules. And as you can see, at the end of this chemical reaction, the purple line is resting at a lower point than it was at the beginning. It went up, but then ultimately it went down lower, which is showing us that the products of this reaction, the glucose and the fructose, collectively they have less Gibbs free energy than the sucrose molecule did at the beginning of the reaction. So here's our little green block, Gibbs free energy. It's smaller than what we had at the beginning. And why is that the case? It's because some of the energy got converted to heat and it just drifted away. It became unusable. So, all in all, what we can take away from this graph, mapping out the breakdown of sucrose, is that there was a decrease in the amount of Gibbs free energy. Free usable energy became less because some energy was released as heat. So here, the starting amount of free energy is demarcated by that top red dotted line, and the ending amount of free energy is demarcated by the bottom lower red dotted line. And the difference between beginning and end is shown here with this double red arrow. Overall, there's been a drop in free energy, which is why this little label here says what it says. This says delta G is less than 0. Delta G is less than 0. What the heck does that mean? Well, the little triangle is the Greek symbol delta, which in science is used as shorthand for change. Delta means change. The G stands for Gibbs free energy. So delta G means change in Gibbs free energy over the course of a chemical reaction. So ultimately, what that little label means is that Gibbs free energy is negative. Gibbs free energy is less than 0, meaning it is decreasing over time. And if Gibbs free energy is decreasing, if delta G is less than 0 for a given chemical reaction, then that means that the reaction occurs spontaneously. So as we see on our little scale illustration, the green block of free energy got smaller. There was a negative change, a decrease in free energy. That means the reaction is spontaneous because if free energy got smaller, it means that some of it got turned into heat, which increased disorder, which jives with the second law of thermodynamics. This type of chemical reaction, a spontaneous one that results in lower free energy, it's called an exergonic reaction. That is the technical term. Now, how to recognize an exergonic reaction from a free energy diagram. If the free energy diagram shows that the beginning level of free energy is higher than the final level of free energy, this means that the energy decreased, heat was released, and the whole thing was spontaneous. And what about the opposite scenario? This whole time, we have been laser focused on this one specific example, the breakdown of sucrose. But what about reactions that aren't spontaneous? Well, let's have a look at a reaction where Gibbs free energy increases. If Gibbs free energy increases, that means that delta G is greater than 0, meaning that there's an overall greater amount of free energy at the end compared to the beginning. The change in free energy is a positive number. These types of chemical reactions do not occur spontaneously because they do not generate heat. So if we look at this diagram right here on the right-hand side, this is how you would map a reaction with an increase overall in free energy. The beginning amount of free energy is down here, and then the ending amount is up here. These types of reactions are called endergonic reactions. So spontaneous ones, the term is exergonic reaction. If it's not spontaneous, we call it an endergonic reaction. What type of chemical reaction might we look to as an example of an endergonic reaction? We were talking about sucrose breakdown as a good example of an exergonic reaction. What is a good real-world example of an endergonic reaction? Well, here's one that you've likely heard of and that we'll be talking about a lot more in Chapter 8, covering photosynthesis. Photosynthesis is an endergonic process that takes 6 molecules of water, H2O, and 6 molecules of carbon dioxide, CO2, and uses the sunlight energy that it captures in the form of ATP to combine all of these little pieces of molecules into a bigger complete molecule of glucose. It basically builds glucose from water and carbon dioxide. So because this process requires an energy input from sunlight, it does not occur spontaneously. It is endergonic. And here's what that looks like on our little scale figure. Photosynthesis starts with the chemical potential energy found in 6 molecules of H2O and 6 molecules of CO2. That's what we represent with our green block on the left. And it adds the energy of sunlight captured in the form of ATP to achieve the final product of glucose, which contains not only the chemical energy from the water and the carbon dioxide, but also the additional sunlight energy added to it. So, as you can see, Gibbs free energy went up. It increased, meaning that the delta G is greater than 0. It's a positive number. So, this reaction is not spontaneous. It requires energy input. To finish off this discussion of exergonic versus endergonic reactions, I just want to give you a quick look at what the free energy diagrams look like side by side for an exergonic and an endergonic reaction. Exergonic is on the left. As you can see, there's an overall decrease in free energy. Endergonic, which is on the right, shows an overall increase. So now we come to a checkpoint. We've talked about the breakdown of sucrose. Let's briefly talk about the breakdown of lactose. Lactose is the primary sugar found in milk. Breaking down lactose brings about a free energy change of negative 8.72 kilojoules per mole. Kilojoules is a measurement of energy. This number, a negative delta G of negative 8.72, this means that the chemical reaction is, fill in the blank, exergonic or endergonic, and that it does or does not, again, fill in the blank, occur spontaneously. So, we now run into an interesting conundrum that may be puzzling you. We have established that the breakdown of sucrose releases heat and therefore occurs spontaneously. We showed this in several different ways, including our scale figure and with our free energy diagram. So if it is spontaneous, why in the world do you not see this reaction happening in your sugar bowl at home? Sucrose, as we said, is regular granulated table sugar. So why doesn't the sugar break down right before your eyes if the reaction is spontaneous? The answer to that question is it actually does. This reaction does happen in your sugar bowl, but it's too slow to notice. Specifically, it would take 440 years for half of the sucrose molecules in your sugar bowl to break down. So naturally, if it's that slow, no one can notice that it's happening. And why is that? Why is it so slow? Well, it's because of this thing right here. This thing that I've just pointed out with the big yellow arrow, this big bump in the middle of the free energy diagram. Remember how I told you that we would come back to this bump and its significance? Well, that time is now. Why does free energy go up first before it slides back down in a chemical reaction? It is because all chemical reactions, regardless of whether they are spontaneous or non-spontaneous, have an energy input required to get the process started. This input is called the activation energy, and that's what this big bump is. It is the activation energy. Think of it as the spark that is needed to get the fire going. A fire is also a chemical reaction that, given the right conditions, will just roar along on its own, but it doesn't get started without a spark. It has to have a spark in order to begin. And so, even though the breakdown of sucrose is a spontaneous reaction, it almost never takes place on its own because it's very, very unlikely for that spark to happen at random inside of your sugar bowl. That's why it takes 440 years for half of what's in your bowl to break down. Now that's talking about your sugar bowl. What about inside of the human body? Things are different in the human body, right? It doesn't take 440 years for your body to extract energy from a can of Coke. If it did, we just wouldn't be able to live and function. It would be impossible. No, it's the opposite. Sucrose breaks down very fast in the human body. So, what gives? How are the conditions in your body different from the conditions in your sugar bowl? The answer is that your body contains these things called enzymes. And those enzymes are the reason why you can break down sucrose in minutes rather than in years. But what is an enzyme? An enzyme is defined as a molecule that catalyzes, or in other words, speeds up a chemical reaction. How does it do this? Enzymes lower the activation energy of a reaction by binding to the reactants and holding them in such a way that the existing chemical bonds can break and new bonds can form more easily. Think about it this way. Imagine a wooden board just sitting there on the ground. You can punch that board 1,000 times, and the probability of it breaking is pretty low, even if you're a karate master. Now imagine that someone comes along and holds the board just right for you to strike it. Now it's much easier to break the board. This is what enzymes do. They hold the reactants in a particular orientation that makes it easier for the bonds to break and for them to be transformed. Of course, this is just an analogy. What it really looks like for an enzyme to do its job inside of a living organism is more like something that you see here in this animation. This is actually an animation that you saw back in chapter 3 when we first mentioned the term enzyme. This animation is showing an enzyme called amylase, which breaks down starch molecules. You can see it grabbing onto the starch chain and facilitating the process of it being chopped up. And so when enzymes do this, they effectively lower the energy barrier of the chemical reaction. They make it so that the spark doesn't actually need to be that strong. That is what we mean when we say that the enzymes lower the activation energy. When an enzyme is part of the equation, your free energy diagram looks more like this, with a little teeny bump instead of a great big one. This is true for every enzyme. This is how all enzymes work. And when I say all enzymes, there are a lot of them. Every living organism relies on thousands of different enzymes to function. Scientists have only just begun to scratch the surface of identifying and studying the massive variety of enzymes that are found in living things, each one catalyzing a unique chemical reaction. A few examples of enzymes related to chemical reactions that we've talked about are the enzyme called sucrase, which breaks down sucrose, the enzyme called lactase, which breaks down lactose, and the enzyme amylase, which breaks down starch. And the bottom line, the key point about the importance of enzymes is that without them, chemical reactions, even the spontaneous ones, would happen too slowly to support life. There would be no life as we know it. Now, when I named those 3 examples of enzymes just now, you may have noticed that they have something in common with each other. I'm going to ask you what that is in a checkpoint here. Amylase breaks down starch, sucrase breaks down sucrose, lactase breaks down lactose. What do the names of these enzymes have in common with each other? And this is a general rule that almost all enzymes follow, as this being part of the name. So when you recognize it, you'll always be able to see when something is an enzyme based on its name. So you have some sense now of how important and ubiquitous enzymes are, and this should align well with the fact that we learned back in chapter 3 when we talked about proteins. At that time, we learned that proteins are responsible for facilitating virtually every process that takes place inside of cells, and a big part of that is because almost all enzymes are made of protein. So back in chapter 3, we learned that proteins are one of the 4 macromolecules alongside carbohydrates, lipids, and nucleic acids. Each of the macromolecules we learned is made of smaller building blocks. And in the case of proteins, those building blocks, those monomers, are called amino acids. And for this next checkpoint, I want you to think back to chapter 3, consult your study guide if needed, and tell me how many different amino acids there are in most living organisms. So now that you've reminded yourself how many different building blocks there are that make up proteins, your memory may also be triggered about how those amino acids relate to each other to form the protein. The amino acids are linked together in a long chain, and the chain folds into this complex three-dimensional shape based on how the individual amino acids within the chain are attracted to or repelled by each other. And the 3D shape created by the amino acid chain is the enzyme. The way the enzyme functions to facilitate chemical reactions is directly dependent on its physical form. And the reason why that is because the folded chain of amino acids creates a pocket in the enzyme called the active site. The active site is the part of the enzyme that binds to the reactant and holds it in place while the chemical reaction occurs. So in the illustration on the screen, the enzyme is shown in blue, the reactant is shown as the little thing in red, and the slot in the enzyme on the left-hand side of its structure is the active site. And the active site of an enzyme is specially made so that its particular reactant can just nestle right in there and be held in place while the reaction occurs. And as I said before, there are thousands of different enzymes in any given cell. And the thing that's beautiful about enzymes is that each one's structure and active site are made to fit the reactant or reactants that the enzyme works with. So, as you can imagine, any mutation that affects the structure of the enzyme, particularly the structure of the active site, those types of mutations have consequences for how well the reactant fits into the active site. This is something that's good to keep in mind when we get around to talking about mutations in our next unit. Another thing that's beautiful about enzymes is the fact that they are reusable. An enzyme isn't just good for running a chemical reaction once, it's good for running it an indefinite number of times. So after the active site is done helping out one reaction, the molecules will leave and the active site will be free again to welcome another set of reactants to do the same thing. So, I have just finished telling you a lot about how enzymes work, and I've also told you how important enzymes are. Not just that they're important, but that they're essential for life as we know it. But that's still kind of abstract. What does that really mean? Why should we really care about enzymes? So, in the next few slides, I'm going to give you some concrete reasons and examples that illustrate why enzymes are so important. Firstly, many inherited genetic diseases are caused by mutations in genes for specific enzymes. One such disease is called phenylketonuria, or PKU. Phenylketonuria is caused by a mutation in the gene for an enzyme called phenylalanine hydroxylase, which is what you see represented here in this animation. And what this enzyme does is it breaks down the amino acid phenylalanine in your diet, the chemical structure of which I've just put in the upper right. So phenylalanine is one of the 20 amino acids commonly found in living things. You get it in your diet, your body has to break it down, and it breaks it down using this enzyme, phenylalanine hydroxylase. But if your phenylalanine hydroxylase enzyme is broken due to a genetic mutation, then the phenylalanine that you eat starts to build up in your nervous system. And over time, this leads to permanent severe brain damage. However, this brain damage is preventable through early detection. It is standard practice for infants born in the United States to be given a PKU screening after birth, which involves pricking their heel to get a blood sample to test for this disease. That's what you see going on in this photo at the bottom. And if a newborn comes back as positive for phenylketonuria, they are immediately put on a diet of special formula that lacks the amino acid phenylalanine, and by doing this, the brain damage can be prevented. As people with PKU get older, they don't have to be quite as careful about avoiding phenylalanine in their diet completely, but that window after birth and during infancy is really crucial for catching this disease and allowing normal development. So understanding how this enzyme, phenylalanine hydroxylase, works how it's affected by genetic mutations, and how to avoid the worst possible outcomes of this disease are all traced back to our knowledge about enzymes. Another reason why we should care about enzymes is that enzymes are crucial ingredients in everyday products that make our lives easier. So, take Lactaid, for example. Lactaid is a pill that people who are lactose intolerant can take in order to be able to temporarily digest dairy. Because the reality is that many people stop producing the enzyme lactase after infancy. Remember, lactase is the enzyme that breaks down lactose, the main sugar found in dairy. And virtually everyone's body makes this enzyme when you're a baby, because when you're a baby, your primary source of nutrition is, of course, milk. But as people age, many people stop making lactase, their bodies stop generating that enzyme, and that makes them become lactose intolerant because their body can't break down dairy sugars. Lactate is a pill, though, that contains the enzyme lactase. So, if you are one of those people who stopped making lactase as you got older, you can swallow a pill that provides you with a temporary source of this enzyme. It doesn't last because it's just a pill. It's not a permanent source that's being generated by your own body, but it can allow you to enjoy that piece of pizza without dealing with the consequences if you are lactose intolerant. Another type of product that relies on enzymes is enzymatic cleaners and detergents. Many cleaners include enzymes that break down oils, starches, urine, etc. I used to work as a vet tech at the ASPCA, and I can personally vouch for pet cleaning sprays that use the enzyme urease. They are great for breaking down urine and getting the smell out of things. And many laundry detergents also contain enzymes as well. A third very important reason why we care about enzymes is that we can kill unwanted organisms by disabling their enzymes. For instance, with antibiotics, which are the medications that we use to kill bacterial infections, most antibiotics are chemicals that disable enzymes that are essential for bacteria to survive. One of the most well known antibiotics, penicillin, works by crippling an enzyme that builds the bacterial cell wall. And without a working version of this enzyme, the bacteria can't build a functional cell wall, and this results in their death. Another example is herbicides. Many weed killers disable essential enzymes in plants. Glyphosate, which is also known as Roundup, is a chemical that cripples one of the enzymes that helps create proteins in plants. And by destroying this enzyme, it kills the weeds to which it is applied. And because of the way they work, these antibiotics, these herbicides, they're considered enzyme inhibitors. They inhibit the function of an enzyme, which then results in death of the organism. But not all enzyme inhibitors work the same way. There are two possible ways that a chemical can inhibit an enzyme. One of these is competitive inhibition, where a molecule enters the active site and blocks the reactant from binding to the enzyme. Basically, it competes with the true reactant to access that special pocket in the enzyme, which then prevents the enzyme from doing its job. But not all inhibitors bind to the active site. Some attach themselves to another part of the enzyme. And when this happens, it is called non-competitive inhibition. So in this process, the molecule attaches to a different part of the enzyme called the allosteric site, which then causes the overall shape of the enzyme to change so that the active site can no longer receive the reactant. And I actually have an animation of this process for you to watch so you can wrap your head around it more fully. So here we have a bacterial cell. And if we look inside of it, here is our enzyme. The enzyme has two pieces. There's the active site and the allosteric site. This is our substrate, also known as our reactant. It comes in to the active site, it gets transformed, it gets released. Now here's our non-competitive inhibitor. It can come in and attach to the active site, the allosteric site, which causes the active site to change. And now the reactant can no longer attach itself to the active site. So that is an example of how non-competitive inhibition works. So this brings us to our final checkpoint of the lecture. We just noted that penicillin is an enzyme inhibitor. I want you to tell me what type based on what you learn here. So penicillin attaches to the active site of transpeptidase, an enzyme that helps build the bacterial cell wall. Without a cell wall, the bacterial cells then can't survive, as we stated. So based on the way it works, does that make penicillin a competitive or non-competitive inhibitor of the transpeptidase enzyme? So we've come to the end of this lecture, but we've covered so much ground in this chapter that I want to leave you with a recap and tell you how all of this connects to what we're tackling next. In this lecture, we have learned that all living things need energy to function. They get it by extracting the potential energy stored in molecules. We also learned that energy is extracted by performing chemical reactions that break the bonds in those molecules. And if these reactions release heat, they occur spontaneously per the laws of thermodynamics. If they, on the other hand, require an energy input, they do not occur spontaneously. The energy input needed for non-spontaneous reactions typically comes from ATP, which is the cell's main energy currency. And one of the main goals of an organism's metabolism is to break down the nutritional macromolecules, extract their energy, and put it in the form of ATP. All chemical reactions, even the spontaneous ones, have an activation energy. That's that hump at the very beginning that makes them slow to happen. So enzymes solve that problem. Enzymes are biological molecules that lower the activation energy and speed up these reactions to a rate that can support life's vital functions. And how does this relate to what comes next week? Well, in our next week's lecture, covering Chapter 7, we will look at how one of the nutritional macromolecules, specifically carbohydrates, is broken down and converted into ATP through a series of enzyme-driven chemical reactions inside of your cells. This process is known as cellular respiration, which is the title of that chapter. And then in Chapter 8, we will look at how carbohydrates are created through photosynthesis, which is also an enzyme-driven process that relies on ATP and energy conversion. So stay tuned for those topics next.